How do I know if I'm going to be healthy over the course of my life? And how do I mitigate that? How do I predict what's going to happen to me? And you can do this again statistically. And it turns out that your zip code is a much better predictor of your health risk than your genetic code. And I'm paraphrasing a very famous quote that it's your zip code, not your genetic code. And that's absolutely the truth. And so I became very interested in understanding what is that other 80, 90% of risk? How do we develop tools to begin to interrogate that information stream? How do we develop mathematical algorithms and computational approaches that allow us to make sense of it? And then ultimately, how do we leverage that information and that knowledge that emerges as a means for drug development and predictive diagnostics, et cetera, et cetera?
Welcome to the Smart Biotech Scientist podcast. I'm your host, David Brühlmann, and today we're diving into the revolutionary world of multi-omics with Mo Jain, who is the founder and CSO of Sapient, who is changing how we understand disease and develop drugs. Mo brings over 20 years of experience in physiology, biomedicine, and computational biology. At Sapient, he's pioneering technologies that analyze thousands of biological markers simultaneously, giving us unprecedented insights into human biology. In this first part, we'll explore how multi-omics is disrupting traditional drug development and why your zip code might be more important than your genetic code, and how mass spectrometry advancements are creating a new era in precision medicine. Let's get started. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Mo, to the Smart Biotech Scientist. It's good to have you on today.
Thanks so much, David. My pleasure.
Mo, share something that you believe about bioprocess development that most people disagree with.
Oh, interesting. That's a way to start here. Get controversial real early on. So what I would say, David, is I believe that drug development as a whole, and bioprocessing being a subset of drug development, is a highly inefficient and flawed system that fundamentally hasn't changed much in the last 50 years. Now, that's not for lack of trying, and it has nothing to do with the brilliant people who are involved in this area. But fundamentally, the way we develop drugs and deploy drugs really arguably hasn't changed in the last 1,000 years, especially from a diagnosis and deployment perspective. And for this system, what we call medicine, to continue into the future, it absolutely must be disrupted.
Yes, there's definitely room for a disruption, and I'm really looking forward to our conversation as we're going to talk about omics. But before we do that, draw us into your story. What first drew you into science, and could you share what inspired you to transition from leading actually a laboratory or a lab at the university now to leading a company? And what were some interesting pit stops along the way?
Yeah, absolutely. I'm still trying to figure out what do I want to do when I grow up, perhaps, David. So I've had something of a circuitous critique career, and that's really a reflection of the fact that I find so many things in life around medicine and science absolutely fascinating, and there's so many very interesting opportunities. So I started as a physician scientist, so I was trained as an MD, PhD, both clinically in adult cardiology as well as scientifically in molecular physiology. And so from the very start, I was in multiple camps where I was practicing clinical medicine, I was doing basic science research. And then for the last 10 to 15 years or so, I really was heavily focused on the research side. And something I truly enjoyed being an academic professor. I enjoyed the environment of academia and the university and the students and the mission. It very much resonated with me. But the challenge or the truth of academia is that there's a very hard ceiling. And academia is not where you go to develop drugs. Certainly, they contribute in a very meaningful way to the drug development and basic science process, but fundamentally, the impact you can have, at least for most academicians. Now, there's a handful across the country who've been able to break through that ceiling, but for the vast majority, the impact you can have on the actual arc of medicine is limited. And that's really what drew me to the private sector and industry, is an idea that you potentially could have far greater reach and far greater impact. Now, as with most things in life, it's circumstance and opportunity and random interactions that catalyze that transition. But this was always a challenge with me, that are you too comfortable in academia? And is it worth finding ways to have greater impact? And that fundamentally is what drew me to the other side.
Oh, wow. What a fascinating story. And what was finally that spark that led you to actually jump over or into the water? Because was it the technology? Was it an aha moment? Was it an opportunity, or you saw the potential, or what was that trigger?
Yeah, fascinating question. So as is oftentimes the case, it was an aha moment mixed with a wow, that's kind of weird. I didn't expect that moment. And so really the long and the short of the story is we were developing technologies that allowed us to deploy mass spectrometry at much greater scale, doing this 50 to 100, 150, 200 times faster than was typical. And as we were going through that process, we began analyzing very large sample sets in the laboratory. And this is when I was an academic. And as this data was emerging, in which we were measuring thousands of different markers in human blood for the first time, and in doing that in scale across thousands of individuals, we began analyzing the data and asking, well, what could we predict about people? Could we tell you who's going to develop heart disease, who was going to develop cancer, who was going to respond to this therapeutic, who was a smoker, who was a non-smoker, what were the environmental exposures that someone had been exposed to, et cetera, et cetera. And as we analyzed this data, it became very evident very quickly that there was a tremendous amount of potential knowledge that laid in these types of markers. So being able to really scale what we measure in humans would provide a tremendous amount of information that wasn't captured by human genomics. So this had been dark to us, and wasn't captured by classic demographics, age, gender, BMI, where you live in the world, et cetera, et cetera. So it was able to provide a glimpse into human biology, human physiology, and human disease pathobiology in a way that had been truly unexpected. And as we were going through that process, it became evident to other folks, and a number of organizations, large pharma companies, large international foundations, et cetera, began coming over to us and saying, hey, we would like to access these technologies. We want to generate this data. Help us understand how our drug works, or who's going to respond to this therapeutic. And that's really what ultimately gave rise to the organization. Now, as is oftentimes the case, we knew very little. We didn't know what we didn't know when we started. We were a group of academics who really, when we spun out the organization, wholeheartedly believed it was going to fail, and failed pretty quickly. But we were going to go through that process. It was in the midst of COVID, so the university and work was largely on lockdown. And we said, well, this will be an experiment. We'll go learn something in the process, and that'll be great. But our expectations for success are quite low. And thankfully, sometimes despite our best attempts, we were very bad at making it fail.
That's awesome.
Yeah. And so that ultimately is what gave rise to our birth and our growth. And as part of that process, we took on a large strategic investment from a private equity founder, and that really allowed us to accelerate quite a bit.
Wow, that's amazing. Wow. I love that you jumped despite thinking you're going to fail, but actually you proved otherwise. That's powerful.
Yeah, absolutely. You have to be okay with failure in this business, or in any business.
Absolutely. Yes. Failure is so important. Yes. Fail often, fail early. That's how you succeed, right? Well, I love talking about this topic because your technology or omics in general has a huge potential. And I worked with various omics techniques during my PhD, also later in technology development. But before we dive into a bit the nitty gritty of omics, can you tell us a bit what is the potential? Because it has enormous potential to revolutionize drug development, disease pathways, to understand how the drug responds, or also how our host cells work, what is the metabolism, and a lot more. So what is the power, the potential of omics, and actually why does this matter in biologics?
Yeah, a great question, David. And so I can reduce this down to a singular number, right? Because in the end, it's really hard to understand impact. You can describe it qualitatively as small, medium, large, but that really doesn't relay the information you're trying to. And so I always like to bring things down to number if we can. So we can think about population attributable disease risk. That is how much of disease risk over someone's lifetime can you explain by current information? And so if we go back 25 years to the dawn of genomics, the sequencing of the human genome, the underlying belief at that time, or the promise of genomics as a whole as really the first omics to be readily scaled to large populations, was that genomics was going to revolutionize human biology, our understanding of disease processes, and subsequently revolutionize target identification, validation, and drug development subsequently. And so with the genomic revolution, there would be thousands of new drugs emerging daily. We would have a precision implementation of therapeutics. We'd be able to tell upfront exactly what therapeutic people should receive, et cetera, et cetera. And that was the great promise. And in no way do I want to minimize sort of the value of genomics, because we've learned a tremendous amount from it. But that promise hasn't largely come due. And we haven't recognized that promise. And you can think about risk in explaining this. And so there are large ways, or there's many ways in which we can begin to evaluate what contributes to disease risk. And one way we can do this is looking at twins, monozygotic twins versus dizygotic twins. And there's large analyses across hundreds of thousands of twin pairs, particularly in Europe, in which you can follow monozygotic twins that share 100% of genetic material versus dizygotic twins that share a portion of genetic material. And in doing so, you can calculate what's the population attributable risk to your genomic DNA. And when you do so, and if you ask most folks who are biologists, hey, what do you think that number is? How much of your risk of developing disease is due to your genome? Or if you ask lay folks, you'll get answers of 85, 90%, 95, 99%. But when you do the math, it turns out to be a lot less. It's somewhere in the order of 15 to 20% is attributable to your genome. And what we know now is from large genomic sequencing efforts through the UK Biobank and many other such studies across the world is that number is actually probably quite a bit lower than 15%. We can only explain a portion of genomic risk in the single digits. And so that opens up the question of, well, what's the remaining risk of human disease? How do I know if I'm going to be healthy over the course of my life? And how do I mitigate that? How do I predict what's going to happen to me? And you can do this again statistically. And it turns out that your zip code is a much better predictor of your health risk than your genetic code. And I'm paraphrasing a very famous quote that it's your zip code, not your genetic code. And that's absolutely the truth. And so I became very interested in understanding what is that other 80, 90% of risk? How do we develop tools to begin to interrogate that information stream? How do we develop mathematical algorithms and computational approaches that allow us to make sense of it? And then ultimately, how do we leverage that information and that knowledge that emerges as a means for drug development and predictive diagnostics, et cetera, et cetera? So the way I think about the multi-omics sphere is we've learned a tremendous amount from genomics about heritability, certainly about the evolution of mankind, how humans have traversed the world where we originated from, population migrations, et cetera, and evolution. And really where multi-omics comes in now is potentially that ability to explain that missing 80, 90% of risk, and in doing so, finally realize the potential of omics data in accelerating drug development.
And what is the state of art now in multi-omics? Because we analyze proteins, we analyze metabolites, lipids, and a lot more different components. But how does it work, for instance, for someone who has never worked with omics? Can you give us just a simple version, and then we're going to dive down?
Yeah, absolutely. So when you think about the canonical laws of nature, how we think the system is organized, and whoever organized it and designed it, I don't know, but the way we believe it's organized is DNA represents the blueprint that gives rise to RNA, that gives rise to protein, that gives rise to protein isoforms, that gives rise to secondary molecules, including metabolites and lipids, et cetera, as well as phenotype and physiology. It's those proteins, those metabolites and lipids that tell your cell what to do, that turn on and off different systems, that turn on differentiation programs, that turn on drug response, et cetera, et cetera. So as you go from left to right, you go from the blueprint to the business end of the system. And as you're well aware, David, genomics, again, was really the first omics to be introduced from a technology perspective that allowed us not only to understand that information stream in DNA in great depth, meaning to be able to go deep and sequence the entirety of the human genome, but also at great breadth and across at scale, meaning that we could do this in hundreds to thousands to tens of thousands to now hundreds of thousands of people. And that both of those aspects are critical. You have to be able to go deep and you have to go far, meaning you have to be able to do this in scale across large populations. Now, the subsequent omics have lagged, but have now finally really come to the forefront, whereby we went from DNA to be able to analyze transcriptomics, initially by microarray and subsequently by RNA sequencing and parallel sequencing processes. And that was the second one. And then most recently, I would say over just the last several years now, we finally have the ability to interrogate the back end of this. So proteins at scale, protein isoforms, or what we call proteoforms at scale, as well as metabolites and lipids, which again represent the chemical business end of this equation. Now, we finally, I would say for the first time, have the ability to not only generate that data at scale, but also analyze that data at scale. So this is a technology advance as well as a data processing and information technology advance. And so using advanced algorithmic work, machine learning, AI-based approaches, we finally now have the ability to make sense of the data at scale. And so this is why there's really an amazing opportunity now that's finally come to fruition. This is really a culmination of technological advances, of biobanking and clinical phenotyping, and fundamentally information technology.
What technological advances are actually making this wide-scale analysis now possible? Is that because of better mass specs, or what exactly happened?
Yeah, absolutely. So you're completely correct, David. So when we think about DNA and RNA, the technical advance had been the introduction of parallel sequencing, what we call next generation sequencing, NGS. And that was really the technological advance there. And so when we think about proteins, when we talk about metabolites, when we talk about lipids, when we talk about these subsequent data modalities, this is really driven by innovations in mass spectrometry. Now, it's not solely mass spectrometry. There have been other technologies that have used binding agents along with DNA readouts, et cetera, but the fundamental leap is really coming from mass spectrometry, the sensitivity of the instrumentation, the stability of this instrumentation, and subsequently the speed of the instrumentation.
When I was working and doing some omics experiments, we very quickly got a lot of information. And you mentioned now we have better technologies to make sense of the data. And that was a big challenge at that time. And I remember even having conversations with my boss. He was questioning whether it was worth the investment to do all these studies, because we ended up with a lot of information, but actually what does it mean? And we were not so sure about it. So where are we there now?
Yeah, you make a great point. I mean, fundamentally, data generation is only the first step, and generating data just fills hard drives. If you don't have the ability to take data and derive information and subsequently gain knowledge and wisdom, you really have not gone the full path. And so this has changed massively. And again, I have to give a tremendous amount of props to sort of orthogonal fields. Genomics certainly set the stage for this. And there's been dozens to hundreds of innovations that have come from handling of large-scale genomic data, everything from how we store data to how we process it, how we make calls around this, so signal processing, and then ultimately how we make sense of this. But also just independent of biology, there's been a massive change in parallel computing, cloud-based infrastructure, and then advanced AI algorithmic work. And all of that, again, comes together at this very unique time and place in history, whereby we can leverage all of this to now make sense of very, very complex data.
And how do we differentiate between correlations and real causality? Because, looking at the data set, you see a lot of parameters going up or down, but is it just by chance? Is it a correlation? How can we now leverage that practically? Or what kind of technology? Is it AI, new machine learning models? Or what are some approaches you have been using?
Yeah, it's a really good question, David. And the first question I always ask is, first of all, do you need to differentiate between those two? So when you think about data, data is only as useful as its use case. And what that means is that you really have to understand how the data is meant to be used, what's the fundamental question you're trying to answer, even before you begin the process. People sometimes are under the impression that you can just make these very large multi-omic measurements, and then there's a magical button that you hit, and the answer spits out on the other side, and nothing could be farther from the truth. I wish it was that simple.
No, it's not that simple. No, definitely not.
It's not that simple, and that's why I always make the point that generating the data, which has been a massive technological advance, really represents step one. You got to get to step 100 before this really does what it needs to do. So the first part is understanding what the use case of the data is. And let me give you an example. For instance, for diagnostic testing, you can make the argument is that causality really the critical factor. If a marker is highly correlated to an underlying phenotype and is highly predictive, that's fundamentally the use case. So an example of this would be HDL cholesterol in heart disease. There's large arguments and many, many different types of biological studies which have suggested that HDL cholesterol is not, in fact, causal for most cardiovascular phenotypes. Now, there's some components of that maybe, but let's ignore that for a moment. But certainly relative to LDL cholesterol or bad cholesterol, good cholesterol, or what we think of as good cholesterol, is not, in fact, causal. But it's an incredible predictor of of someone's risk of heart disease. So it still has an incredible use case in understanding risk prediction and for diagnosis of disease and prognosis, even though it's not a causal agent itself. Now, the fact that it's not a causal agent makes it a bad drug target, and that's a whole nother issue. But correlated markers can still be quite important. So you asked an important question about how we begin to understand causality. And this is where fundamentally intervention studies are still quite important, where you have to be able to go in and do fundamental biology. And the way I think about this very complex multi-omics data is that the goal of it is to identify in the end singular targets or singular mechanistic pathways that you then do experimental work on. Now, certainly there's a number of computational approaches that we can use to infer causality, Mendelian randomization, et cetera, et cetera. So there's a lot of statistical tools we can use to provide some clues about causality. But in the end, particularly in the context of drug development, one needs to go do a fundamental experiment where you somehow perturb that protein or that gene or that biological pathway, and you see a secondary consequence of that perturbation that suggests that it's going to be a good drug target.
Yeah, you're making an excellent point, and I want to just stress that once again. Before starting, you need to know what is the end. What actually do you want to describe, or what is the thing you're studying before actually generating tons and tons of data? And I love also that you mentioned in certain cases, actually, a correlation is fine. Yes, the cholesterol is a great example. Absolutely. But I'd say in certain cases is when you're studying metabolism and you want to understand actually why this happens, then you have to dig deeper and really to understand what is really correlated.
That is absolutely correct. And I'm not sure multi-omics or any singular omics by itself allows for faster knowledge. But what they allow us to do is to look at a much wider lens. So as opposed to studying one thing up front that we think we know is the answer, we can ask many questions simultaneously and then fundamentally prove it. Now, you could argue that time scale is maybe a little bit faster, but the great difference maker here may not be the time, but rather the certainty in the result. I'm not predisposing or presupposing that I know the answer. I'm letting the data tell me what the answer is.
That brings us to the end of part 1 with Mo Jain. We've just scratched the surface of how multi-omics technologies are revolutionizing our understanding of disease pathways and drug development. In part 2, we'll dive deeper into the practical applications and explore how biotech scientists like you can leverage these powerful tools. Before we wrap up, I want to remind you that I offer free consultations to answer any process or technology development questions you might have. If you're struggling with bioprocess challenges or looking to optimize your development workflow, I'm here to help. Just visit BrühlmannConsulting/call and schedule your free consultation. Stay tuned for part 2 with Mo Jain, where we will explore even more practical applications of multi-omics technology. See you then. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.